Welcome everyone to our Boeing Distinguished Colloquium. It's a huge pleasure to introduce Adjunct Professor of Applied Mathematics and also Professor of Neurobiology and Biophysics, Adrienne Fairhall. This is an applied math talk, so I wrote a multi-scale introduction here. We're going to go local to global, and we'll start locally. For the last 20 years, Adrienne has defined the field of computational neuroscience in Seattle. So that started with the first Seattle-based, but really the first global also MOOC in computational neuroscience, which is registered up to 100,000 learners. The very first national training program, federally funded in computational neuroscience, was brought from the nation to here by Adrienne in 2011. And in the 15 years since, there have been millions and millions of dollars that have all been funneled to external groups outside of her own through the programs that she's led, including now an even larger program from the Shanahan family, which supports research between the Allen Institute and the University of Washington. And locally here in town as well, Adrienne, as you know, worked with this department as well as neurobiology and biophysics and engineering to co-found the Computational Neuroscience Center. Outside of Seattle, so one step larger in scale, Adrienne has directed and founded the premier international courses in computational neuroscience in Boston, in Cape Town, South Africa, and in the Pacific Northwest. Serves as a trusted advisor federally to the NIH at the very highest levels in the nation, but for me what's more special about that is about the most difficult issues there are. And these are issues that involve ethics, experimentation, and the future of the field. There are very few people who are willing to step up, do the targeting that produces, and Fairhall led the nation in that field. This, as well as much more importantly, her science, which I'll talk about in a second, was recognized by her election to the National Academy, the biggest scale, here we go, almost done, I promise, is the scale of ideas that transcends really scale. As many people know, Adrienne is a dynamicist at heart. Her PhD work was in theoretical and statistical physics at the Weizmann, here we go, KK, in mathematical models for high-dimensional turbulence. And she brought that focus on nonlinear dynamics throughout really her whole career, but became famous internationally for building the link between nonlinear dynamics and something new. And that something new is coding and computation in biological systems. An example of her beautiful work is to exploit the nonlinear dynamics in single cells through asymptotic expansions and stochastic differential equations to show how particular types of nonlinear dynamics can perform and produce, for example, scale invariant computation, a theme of my introduction. And today you will see how that perspective on nonlinear dynamics scales up to networks as well. So with no further ado, our very own, at least shared, Professor Fairhall.
Thank you, Eric, for that incredibly kind introduction. I hope you all agree that our joint effort recruiting Eric here is one of the best things your department and I have ever done. I take the part of having taken him to Vivace, which I think was a key part of the recruitment. So I have a lot you can thank me for. As Eric said, our lab has worked on many things over the years, and today I'm going to focus on a strand that's kind of run through our work over the past 10 or 15 years, and this might be a bit of a smorgasbord of some examples of specific studies that we've done, primarily on the topic of birds. Just wanted to zoom out momentarily because in case you haven't been to a neuroscience talk for a while, just wanted to remind you why this is such an exciting time to be in neuroscience and involved with neuroscience. We are really bearing the fruits and the benefits of the investment of Obama in the BRAIN Initiative, which has brought incredible developments in many areas that have really transformed the field of neuroscience. High throughput recordings, both optical and electric, electrophysiological, as you see here. This is an example of a optical recording. This is an example of the kinds of data that people are now routinely taking in their lab, where they're able to record from hundreds or even thousands of neurons across the brain simultaneously. amazing work going on at the Allen Institute and elsewhere in decoding the connectome. We're starting to really have the underlying wiring diagram of the brain. And also, of course, in AI, right? AI methods and tools that are allowing us to extract information about behavior, in this case, of this package, and aspects of the statistical structure of neural signals at a resolution and depth that we've never had before. All that said, many people can tell you about their amazing advances in all their areas. We're not there at the moment with birds. Just a little heads up. So birds are an amazing system for the topic that I'm going to tell you about, which is learning. And I don't, despite that introduction, I don't want to disappoint you by the scale of the data and the sort of precision that we're yet able to achieve with birds. Partly that's because birds are finicky, right? So they sing. It's a beautiful behavior that they're compelled to do, but they don't like to do it with an electrode in their head. And so there's some limitations to moving into the situation of large-scale recording. But I hope at the very end of the talk, you'll see an example of what we're starting to do with some pretty decent recordings from some of the networks in birds. So birdsong is really a canonical learning system. I just want to quickly introduce you to all the people that have played a role in our studies of this over the years. Alison Duffy, who was a PhD student and has stayed on in the lab, and cast of characters that you'll see show up as we traverse their work. Songbirds are trial and error learners, right? There's very few examples that we have such a tidy example of trial and error learning. And what is beautiful about the particular species that we study, which is zebrafinch, they're an Australian native, and they sing one song. So that bird will learn its song over time, from childhood onward. And in the beginning, it kind of babbles. The song gets better and better until it crystallizes. And what you're looking at here is sort of a snapshot of renditions of the song. Every point represents the distance from that rendition in some space to the final song. And you see that they're generating quite variable renditions. And this is kind of over the last phase of the crystallization of the song. You see that it's kind of getting better and better over the course of those days. But even here, the song is still quite variable, but reasonably stereotyped. And the fact that it is singing this single song means that we can even make a plot like this, where we can measure the distance from one rendition to its kind of target. So that gives us really a lot of power in trying to understand how it is doing that learning. So I'm going to show you this one picture of the brain. It's the only sort of neuroanatomy kind of picture you're going to see, but I just want to point out that there's a very particular way that bird brains are organized that's a little bit different than the way mammalian brains are organized, which is that the relevant regions, the kind of functional areas, rather than being kind of patches of the smooth surface, as we have in our brains of the cortex, they are instead nuclei. They're kind of round. And so they're well-defined, functionally well-segregated. And in the case of birdsong, those regions are dedicated solely to the purpose of learning and producing the song. That's an unusual situation when you think about how mammals generate movement. We have our thalamocortical basal ganglia circuits that do that learning. But of course, the motor patterns that get generated are a mixture of all kinds of things, right? Movement is a very high-dimensional space. When animals learn to move, they learn to move all of their limbs. In the bird, the regions that are specifically targeted at the aspect of movement that's involved in song are well delineated and segregated. And so in some ways, this makes, the fact of this single unitary song, this well delineated architecture, and the trial and error nature of the way that they learn. I like to think of the zebrafinch as kind of the hydrogen atom of motor learning, right? It's just perfect, simple example. All right, so the regions we're going to focus on are these two, HVC and RA, and then these three. I'm going to call it LMAN, Area X, and DLM. You will see those names a lot, and I'm going to make this picture a little bit easier to process by putting it in this form, which is also much easier for me. So what we're looking at is a system in which we have two nuclei that are primarily responsible for generating the song. HVC is a premotor area, RA is a motor area. Those neurons eventually go to the syrinx, which pulls the muscles and generates the song. This group of nuclei is called the anterior forebrain pathway, and this is very important in learning and memory. And this region is the VTA. Mammals also have a VTA that is responsible for generating dopaminergic signals that we think are very important for evaluating how well you perform. So this whole system is critical both for the original learning of the song and also for maintaining the song. The bird actively maintains its song throughout life. If you deafen it, it will deteriorate, so it's doing it in a way that requires auditory feedback. At the same time, it's a system that's incredibly robust to perturbation. I'm going to show you an example of that. So altogether, because it's a trial and error learner, reinforcement learning is really a very natural way to think about what the bird is doing, right? So how does reinforcement learning work? I'm not going to give you, for machine learning people who are hoping for something a bit more sophisticated, we're really working with the most bare bones version of reinforcement learning. The way we implement that is that there's an actor, right, that generates some action that interacts with the environment. Here we don't really have an environment. The bird just sings a song. Given how good the song is, right, did I turn that off? There will be some reward, right? It will do some internal evaluation of how good that song is. And then in reinforcement learning paradigm would then calculate the reward prediction error. So you'd see what the difference is between the reward that you got from experiencing the action that you took and your internal notion of how valuable that action is, so what you were trying to do. And then you'll use that difference to update your model for how valuable a given action is. So this goes on, right? So you generate actions, you have to search around in action space, and on every trial you'll evaluate how well you do and then update your actions in order to minimize your reward prediction error and sing the best song that you can. So we think about this part, right, the actor, as sort of an experimenter, right? It's generating some suite of actions that are its attempts, right, to do the right thing. How does that map on to this song system? So I told you before, we have these two regions in red, this motor pathway. So we can think about that as the actor, what is actually generating the song. These regions in blue, they have been traditionally thought about as sort of the experimenter. What they're essentially doing is injecting variability into the song on every trial. You saw from the original plot that I showed you that there is still a lot of variability even when the song is finally crystallized. So this set of nuclei are responsible for that. You see that there's one nucleus here, LMAN, which is actually projecting directly into the motor pathway. That's carrying essentially a noise signal that's generating that variability from trial to trial. And then finally, we can think about a VTA, which is also true in mammals, as sort of the critic, right? It's monitoring what's going on and giving some estimate of the error or the reward prediction error. All right, so what I'd like to go through with you is how this network works at a circuit and a neuronal level to implement this reinforcement learning algorithm. You can see there's lots of moving parts here, so I'm going to keep trying to use this diagram to keep referring you back to what these individual regions are doing. So here's the overall plan. First, we're going to look directly at HVC. It has very special dynamics that are quite beautiful, and we have an active collaboration that we've been working on for years about how that region itself works. And I'll show you how some models of how we think it might be doing kind of self-organized learning and maintenance in order to generate the dynamic to underpin the song. We'll zoom out a little bit and think about some classical minimal models for how this circuit is doing reinforcement learning. We'll turn over to this part, right? So this is the critic. So is the critic real? I'll show you some data from some recent experiments that reveal to us to what role it really is playing in this this potential paradigm. I don't know if I'm doing that or if it's just going up and down all by itself. So apologies for the variation there. And finally, we'll think about this experimenter. What does it mean to be an experimenter? How can a set of nuclei, generate randomness if they do? All right, so let's start here. Self-organized learning and maintenance. So area HVC, right, is the area that sort of organizes the song, and it's really has a very beautiful firing pattern. So this is what's going on in HVC. So what you're seeing here are raster plots of the neurons, or individual neurons firing in HVC. So a raster means that what's happening here is we've lined up, or Richard Hunloser lined up his recordings from individual neurons as the bird sang multiple renditions of the song. So you just stack them all up for each rendition. And so what you're seeing is that each of these neurons are firing extremely precisely, right? So this one neuron fires once throughout the song at this very particular time. And so there's a whole sequence of them that fire. Other work has shown that the firing of individual neurons sort of tile time throughout the song, and each of them fire very precisely. What happens downstream, this region that then drives the muscles in the syrinx, right, that kind of pulls the strings of the actor, these now, as one might expect, fire multiple times throughout the song. So if you imagine that what you have here at the top is sort of like a metronome, right, that's keeping the time throughout the song. What you have here is kind of how that metronome is then pulling the strings of the muscles that then generate the song. So if we imagine that this neuron is pointing to some particular muscle, any individual muscle, there's a small number of them, maybe 13, are implicated in multiple syllables of the song. And so you should see, right, that this neuron is firing multiple times as that muscle is participating in multiple syllables as the bird is singing the song. And here you see the spectrogram, right, of the song itself. So clearly, right, the learning problem is to tie time, as expressed here in HVC, to motor output, as expressed in these RA neurons that then drive the muscles of the syrinx. So what happens in HVC? So just to cartoon this, right, what we're looking at is the excitatory neurons in HVC. A bunch of them fire at a given time, and then another bunch, and then another bunch, and then another bunch. Very importantly, the ones that are firing together are not next to each other. They're kind of distributed throughout HVC. So it's not something as simple as just a slow-moving wave, right, that's passing through HVC. Instead, it's groups of neurons that are firing simultaneously. And in the background, you have inhibition, right? So there are inhibitory neurons as well in this circuit, and they form kind of a global background of inhibition. The way that it has been demonstrated that this system works is that the sequence is generated within HVC. So it really is a kind of chain of firing where one group of neurons drives the next group of neurons, drives the next group of neurons. There's a little tweak to that, which is that it's polychronous. What that means is that one neuron can drive another neuron if it's the next element in the chain, but how long that element is depends on how long it takes for a signal to get from one neuron to the next. And so there are temporal delays as the signal travels along the axons. That means that you could be, say, 2 time steps back and still drive the next neuron in the sequence if you're far enough away, right? So the signal takes that extra time to travel. So what's the implication of that? What do we expect from the connectivity matrix? It should look something like this, right? So neurons are connecting just off diagonal to the next one in the chain, but it has some width, right? Because you can be connecting to the next plus one if you have a delay that's appropriate. And these are the connections to the inhibitory neurons. They're kind of global. And what that sort of global inhibition is doing is essentially winner take all, so that when one group of neurons is firing, the inhibition kind of pushes down the activity of all the others so that only one group of neurons is firing at once. So that kind of enforces the sparseness of the activity in the matrix. So as I said, this sequence is fundamental to song. What happens if we disrupt HVC? So this is work that we've been undertaking in collaboration with the Lois group, Carlos Lois at Caltech. What Carlos's lab did was to silence about half of the excitatory neurons in HVC. And so this was what happens to the song. So the bird is singing the song. Before the perturbation, you infect it with a virus that silences half of the cells, and the song essentially goes back to the babbling stage that the bird was at before it really learned how to sing properly. And so the song falls to pieces, it becomes very disorganized, becomes temporally very disorganized. If you wait, though, for a couple of weeks, even though these neurons are permanently silenced, the song will restore itself, right? So if you look at the spectrogram there, 14 days later, it's pretty much identical to what it was before this perturbation. So that's an amazing recovery from a very dramatic perturbation. Yeah?
Can you say again in terms kind of the connectivity matrix what the perturbation is?
Yeah, destroying half of these green guys, or silencing them, right? Preventing them from speaking to their neighbors.
And what are the matrix of the green guys?
So, sorry, yeah, I didn't keep the colors consistent. Blue is inhibition. So the blue here are representing inhibitory neurons, which are connected to many neurons kind of at random. And then we have this more detailed chain of connectivity among the green neurons that are the ones that are carrying the sequence. What I haven't shown you here, but you could have integrated if you watched that, is that there are actually many neurons in this network that are silent, many of the excitatory neurons that don't actually fire during the song. And that's going to be important for what we're going to see later. All right, so it can recover after 14 days. So this is I just love showing this picture because it's so beautiful. So, Alison, who's at the back there, took the songs, right, the spectrograms of the songs, and projected them into a low-dimensional space using UMAP. And it's just a lovely way to see what's happening to the song. So this is the song rendition, sort of expressed in this low-dimensional embedding. And now you can see what's happening over the subsequent days after this perturbation, right? It's a total mess. None of the syllables look like they did before, but over time, right, the song starts to re-emerge from that noise, and eventually you get back something. You can see that there are tiny details that are a little bit different, but essentially the song is back to what it was before. Eric.
Very quickly, on the recovery, did the neurons sort of come back into this circuit, or are they all gone?
So the trouble with this experiment is that what we have is behavior, and then we have slice recordings. They have verified that neurons that were silenced do not come back, right, over at least 3 months. So over this time course, they don't restore themselves. But it's a very interesting question, what exactly is happening. I'd love to be able to show you just a movie of what was happening in the circuit over the time course of recovery. We're hoping to be able to do that. The lab now has that technology, but we didn't have it at the time of this study. So just to show you how that recovery goes, right? So how can we quantify it? We can look at individual syllables, and now we take those spectrograms, project them in a low-dimensional space, and so according here to their first two PCs, these are different syllables. Here's the original syllable, and then as it's perturbed, what we do is we try to identify which are the points that are kind of closest to that original syllable. And then we quantify how far away they are, right, from the syllable prior to start. And so we can look at the distance from the original syllable, and you see, initially, there's always some distance, right, because there's always some variability in that blob, right. So individual songs have some distance from the cluster center. So there's some intrinsic variability. That's our background, and then that distance grows, and then it recovers back. Generally, not so much in this, but generally what we find is that even the amount of variability in the song often recovers to very close to what it started before. The key thing, and what is sort of fascinating, is that one might think, right, the bird is basically back to childhood, and it's using auditory feedback to relearn the song, using all of the machinery of reinforcement learning that I showed you before. That turns out to not be true, because if you stop the bird from singing, it ends up recovering on exactly the same trajectory. So you silence the bird by, if you put a kind of vest on them, they hunch over and they just don't like to sing. And so they sing maybe 2 or 3 times over the course of several days. Yet, after that time, the bird is just where it would have been if it had been able to sing throughout. So the conclusion that we draw from that is that this region, HVC, which is critical for the song, is somehow able to recover itself, right? That it's able to restore that sequence even in the absence of any singing. So it's not that it's taking the ones that are there and kind of rewiring them so the song comes back over time. We must need to do a little bit of that tweaking at the end, but largely we think that it's able to rebuild the sequence inside that network. And so this is part of the thesis work of David Bell. He did the painful work of building this model and then testing what kinds of plasticity rules could make that happen. You know, what works to bring about the changes that we saw. And so we take the model, we perturb it, right? So this is the question, you know, by silencing a large fraction of the neurons, and then we try various plasticity rules and see which of them account for the data. And the data consists of two things. It consists of the song itself, but we also have intracellular recordings in slice that tell us how much the synaptic weights changed in the slice. I unfortunately don't have slides of that data, but that's kind of a key piece that we're trying to capture as well. So what are the typical plasticity rules that neurons have available to work with? So there's two key types of plasticity that are intrinsic and generic in biological neural networks. One is homeostasis, right? So neurons have the capability, if they say their firing rate goes down because they lose some inputs, they have the ability to kind of dial up the weights of the inputs that they're getting in order to restore their firing rates to what they were before, right? So they have this kind of internal knob that they're able to upregulate, right, the weights of inputs so that they can fire like they used to. So that's one type of plasticity, homeostasis, which seems like a very natural one. And another type of plasticity called spike timing dependent plasticity. That is for a neuron that's getting some input, the timing between any input coming in and the time that it fires, there's a plasticity rule that will regulate the strength of the synapse according to that timing. So that's another basic kind of plasticity rule that biological neurons have. So if we apply those rules, right, so here's our pre-perturbation sequence, and here we've just chosen at random some neurons from the circuit, so we don't necessarily have everyone in the link. After recovery, you see that there are neurons that are still firing, you know, in that sequence, but there's a big difference in the way this recovered network works and what we might like to see, which is that, of course, the neurons that are silenced aren't there anymore, and so there are just missing links in the chain, right? The overall strength of outputs coming out of HVC that are needing to drive downstream RA is just much lower in volume, right? The number of outputs is reduced by a factor of 2, and none of these plasticity rules can cure that problem, right? So what we did was propose a different kind of rule, a novel rule, but one that we think is biologically plausible, where this provides the network the opportunity to kind of know what's going on in its neighborhood, right? So individual neurons, if you're silent, homeostasis, you can upregulate your neurons, but if you never were firing before, none of these rules will help you to fire in the future, right? You weren't part of the sequence before. You can't be part of the sequence according to these rules, right? There's no way to bring you in. What we wanted was to propose a rule that would help to actually make use of that pool of neurons that are there and weren't previously part of the song. And one way to do that is through what we call a population homeostatic rule. So we propose that there's a mechanism by which a neuron is able to kind of tell how active its local population is, and that upregulates the firing or the sensitivity of neurons in the network such that some of those previously silent neurons are able to be brought in, right? So after the perturbation, here's the idea. We imagine that there's some locally secreted factor, right, that has a concentration that depends on the amount of activity. If that is knocked down by reducing the total activity, then there's a mechanism that is sensitive to that external factor and will upregulate the activity of neurons in the network to recover that level of this factor. Right, so what that says is that the local network is sort of able to communicate how active it is at a kind of larger length scale than just the single neuron level. So this works, right? So what this does is introduces into the chain networks that were previously silent, and it also, although I'm not going to show you the data, it captures the pattern of synaptic strengths that we saw in the data, right? So there was some upregulation of weights that was anomalous and that wasn't captured by the plasticity rules that we tried first. This plasticity rule is able to account for those synaptic weight changes. All right, so what, we like that, it's cool, right? We kind of developed or proposed a novel kind of learning rule that's kind of now handing it back to the experimentalists to see if they can find evidence for that kind of learning rule. But I'm going to take you down David's intellectual pathway, which is that over the course of trying all these many different kinds of intrinsic rules and trying to tune them to see if he could capture what was going on in the population, he got very frustrated at kind of the ad hoc nature of what he was doing. There's many learning rules in the literature. They have lots of different parameters. Which ones should he use? You know, he could cycle through many variants of their parameters. And that was frustrating to him. And so what he did was to consider whether there might be a more efficient way to do this, right? Are there rules that in general will build a sequence for you in a kind of unsupervised way without you having to introduce any special pieces to it? And so what David did was to turn to this interesting recent literature on metalearning, right? Can you use a way of thinking about the space of learning rules to kind of discover in that space a subset of learning rules that if you just put them into your network will just automatically build sequences for you in a constant and unsupervised way. So that's what he did, right? So he here's the trick, right? So you choose a basis of forms of learning rules that involve the presynaptic activity, the postsynaptic activity, the current strength of the synaptic weight, and some time constant. You might think that there's sort of a kernel over the recent history of these factors. You implement them in your network. You evaluate loss, which you define via what you want the outcome of the network dynamics to be. And then you search around in the space of these parameters to minimize that loss using a genetic algorithm. And so when one does that, so just to show you what the rule basis looks like, so we use this polynomial basis of weights, pre and post synaptic activity, and then a kernel over the recent history of the firing rates pre and post synaptic with a time constant tau. So here's the way that we set up the kind of the cost function. So what we wanted this network to do was to generate sequences. And so one way to demand that is to demand that you would like to be able to decode time, right? Because remember what our HVC network is doing. It has a sequence that has the role of encoding time in the song. So that's going to be our figure of merit. So initially we drive some subset of the neurons in the network, so you get these firing patterns. And now we ask that at every moment in time, one can decode the time, right? And that's our figure of merit. And now you let the algorithm run, right? And what you find, so here's the initial conditions. So initially you have some firing patterns that are kind of random, but over time, as the network converges, you end up with this final sequence. And so what does the weight matrix look like after this learning is done? It looks like what we would hope, right? It's this sort of polychronous, sort of broad, diagonal yet a little bit broad network that has the form that we would have expected from this kind of chain model. So what was the rule that was found, right? Ideally, what would have been nice is that there's some unique form, right, to a rule that was specified with just a couple of parameters. That would have been great. But what we find here, what you're looking at is the coefficient of the different terms in the rule over time as the genetic algorithm is running. And what you see is that there's actually lots of coefficients that are far from zero. So how do we kind of try to reduce this space and make sense of what's going on? So what David did was instead to see if we leave terms out, right, if we drop them, by how much is the loss changed by dropping individual terms. And then you can take it the other way. You can take these terms that cause a lot of loss, right, the ones that cause the most dramatic difference in how well the learning rules work, and then put them back in one by one. And so what that leaves us is an approximation for the rule. And it turns out, so here's what that approximation looks like. Our meta-learned rule has this form. Right, so it depends on the pre and post synaptic activity with some time constant, and and also the pre- and post-synaptic activity, where you now integrate over the post-synaptic activity over some time scale, and then a term that also depends on the weight between them, right? So it has these three terms, which if you look back here, so some of you will know Euler's rule, which is a way, a standard rule to wire up networks. It looks very similar to that, right? This has a lot of similarities, right? This has a little bit of integration over some window of time, but it's essentially the same term. And it's xi, here we have something that just has a little bit of temporal kernel included. So essentially what we found is sort of a generalization of Euler's rule. All right, so then next thing David did, that was all just including plasticity between the excitatory neurons. What happens if you also include plasticity between the inhibitory neurons? This is computationally quite a pain, because now there's lots and lots of terms. But what David found, just to quickly summarize, was that you end up with networks that are a bit more general, right? So this was just excitatory rules only. This is if you include inhibitory rules. So you get sort of slightly tuned, right, inhibition in this network. But this network that has the rules operating on the inhibitory neurons has the nice property that it's actually a more robust network than the pure excitatory one, or the one that has the learning rules only on the excitatory neurons. So how he tested that was by adding noise, adding synaptic noise, by kind of turning over synapses, right, at a regular rate. And here's how the error or the loss scales for the networks that only have plasticity operating on the excitatory neurons. And here's what happens if you include it also on the inhibitory neurons. So you end up with a network that is much more robust to that kind of synaptic noise. All right, so what I've shown you is that we could discover rules that build self-sustaining and self-healing sequences. So it's nice to imagine, right, that maybe this is the way that HVC, if it just was genetically set up, right, with learning rules like this that are operating all the time, that would allow it to build the sequences that emerge over development and to maintain them throughout its life and to keep them nice and precise, even in the presence of synaptic noise and synaptic turnover. We would now like to understand if this accounts for recovery under targeted perturbation. So with Carlos's lab, we're now looking at some experiments in which they don't just kill the neurons, for the whole time or silence them for the whole time. They instead add temporally precise perturbations at a particular time in the song. And we see already, what we see from their data is that does throw the bird off, right? It sings the song, and on some large fraction of the trials, the song gets perturbed. But over the course of about 3 days, it's able to completely get rid of that perturbation and sing the song perfectly, even in the face of that perturbation. So we'd like to understand how that could happen and how that might happen if now we tweak parameters of the perturbation. You only do it half the time. You put in some catch trials. So do our rules work to solve the problem? What might be going on in HVC to deal with those perturbations? David has gone on to ask if there are other kinds of dynamical motifs that can self-construct. And I won't tell you about that, but I would love to invite you to his thesis defense, right? He's about to present, and we'll be giving a talk about that in a week or two. All right, so what we've seen, we've just spent quite a bit of time on how this initial timing gets set up in HVC. Now what we'd like to understand is how that gets wired up, right? So the fundamental learning problem that this system has to solve is how to know which muscles to pull at what time. And so this HVC to RA synapse seems to be the critical one, right, where learning has to happen. So what we'd like to have a look at is how people have thought about this learning in the past, right? So there are some classical models that really just propose that this is a simplified reinforcement learning paradigm, right? So there's this work from Doya and Sinofsky where what they did was treat this as a kind of 3-layer network where you have this timing coming from HVC, you have your output coming from RA that then drives your motor output. And in the middle, you have this perturbation coming from LMAN that just sort of acts like a random number generator, throwing variability in, and you then choose, right, the examples that happen to lead to a better song, and you strengthen them, and that's how you converge. This was implemented with a bit more precision by Ila Fiet, working with Sebastian Sung, where they kind of put a bit more flesh on those bones. So again, same kind of idea, right? You have RA generating the output to the song. You have some critic, right, that's operating at these synapses. L-man generates perturbations. Depending on whether those generate a good change or a bad change, this critic will then regulate the plasticity that then changes the HVC to RA synapse, right? So these are called empiric synapses. So empiric meaning they're kind of sampling, right, testing the space of possible motor outputs. And these synapses from HVC are plastic, and their degree of change is sort of guided by the L-man input. So yeah, these are basically variations of the reinforced algorithm, if any of you have heard of that, from 1992. So we played a bit with that model, and this is more kind of in the spirit of history. So we applied this model to account for something that we have seen also in data, again from our collaborators, and that is that given that the song is built on this scaffolding, right, of timing, then one really wants that timing is very reliable. Right? One would like that every time the bird sings a song that you get exactly the same firing out of those neurons. As you saw from those brief experiments, that's true over the time course of 10 trials. Is it true over very long periods of time? Data that Tim Gardner took suggests, so this is using optical imaging, looking at neurons firing in HVC over very long periods of time, seem to suggest that there are some fraction of neurons that actually change their firing over time. They stop firing, or some start to fire at a different time. And so how does the whole system work when you have a low degree of variability in how the HVC neurons are firing? Because that should kind of mess up your algorithm. So what we did, and this is work with Elliot Abe, who is now a postdoc here at UW. He was working with us, was one of our original cohort of our training grant. He worked in my lab as an undergrad and did this work when he was an undergrad here. We implemented this model, and then we asked what happens, right, when you now perturb the firing of the HVC neurons. And so what we did was to add in more and more perturbation and see what that did to the song. So you can change the number of times. So here you can see what's going on, right? So if you look at the red trace, that's how well the network trains, right? When we set it up, as I described to you, it trains and then it keeps a very low level throughout. If we now add these perturbations where we kind of scramble the firing of some small number of neurons, then the error, of course, jumps up, but then it relearns and it will relearn and relearn. But you see that the amount by which it relearns always stays a little bit above the best possible case if you had no perturbation. And so here's the error improvement. As you increase the number of perturbations, you see that the amount by which you improve, right, sort of you take a bit of a hit by adding this perturbation. What's good about it, though, and why the bird brain might actually do this, is that it actually puts the network into a state that's sort of favorable if you ever want to change your song. Right? You could imagine that as you get older, or many of us can easily imagine, don't have to imagine, that you don't quite control your muscles in the same way. Your legs get sore, you get a bit fatter, right? You need to change the way you implement your motor control. And so a little bit of ongoing plasticity is a good thing. And so if now we take the network and we ask it to produce a different song, if we've trained it without perturbation, that is very difficult for this network to do. So here's what now you're seeing how that relearning error decays, right, as a function of the number of training songs. And so it decays away. These different curves are how rapidly it decays given the number of times that you've perturbed it during the original training. So as you increase that number of perturbations, the amount of time, it doesn't look in this plot as though there's a lot of change, but if you actually calculate that decay constant and you look at it here, you see that there's actually a 50% improvement in how rapidly you can learn a new song if you have trained it in the presence of perturbations in the first place. So it's not just quicker to learn changes, it's also more robust to network damage. If you knock out a couple of these neurons, and you look at the amount of error that causes, right? So here's the original network. You break something, right? You ablate some of these neurons. You look at the error. If you have trained it without perturbation, you get a huge, degradation of the performance of your network. The more you perturb, the less damage that does, right? The less bad that kind of ablation is for the performance of the song. Why is this? I forgot to put the picture in. I meant to do that at the very last minute. Why is it? It's because when you train the network without perturbation, you generate really strong perturbations in the WIJ matrix. You have this very strong timing signal. You're driving this downstream output. That timing and the fact that the learning kernel, right, has some temporal width means that subsequent WAJs, right, when one neuron is projecting to a set of downstream neurons, they become extremely correlated, and you end up with a matrix that is kind of overtrained on this very specific sequence. And by scrambling that sequence from time to time, you end up in, you can think about that original training as sort of leading you to be in a network that's sort of sitting in this deep potential well of very specific solution that now if you want it to retrain to do something else, it's very hard for it to get out of that well. Whereas if you put in these perturbations, it's really very much like node perturbation in training of an RNN or any kind of neural network. You end up with a much more generalizable solution. And so that kind of dropped out of here, and I think gives a really compelling idea for why this intrinsic variability might be there in HVC. All right, so one of the issues that some of you might have spotted with why this picture that we've always had of how the birdsong circuit works as a reinforcement learning problem can't be quite right? So we've said that what we want to do is wire up the timing in HVC to the output in RA, and all of the models that I showed you suggested, oh my God. Okay, we're about a third of the way through. So where we want that error to be is here, right, at this transition point. But where that error is actually coming in is in a totally different part of the circuit, right? So how can we be sensitive to error here in our training system when we're getting error here, way over the other end of the circuit? So hopefully, in this last 10 minutes, I'll very quickly tell you how we think that might happen. Okay, so first, let's just have a look at what signals are even being conveyed by VTA, right? Is it actually behaving like a critic? So we have good reason to think that it is in fact carrying error signals. So this is an experiment done by Vikram Gadekar, who's still a collaborator of ours. Now he's at Columbia. What he did was to take, while the bird was singing, he added a perturbation at a particular time during the song while recording from the VTA projecting neurons. So he was recording their firing rates so he could tell, right, how much they were driving this dopaminergic signal that was going into area X. And on about half the trials, he put this distortion in. On the other half, he didn't. And what he saw was that there are neurons that were modulated at that moment in time, such that when there was a distortion, they dipped, and when they were not distorted, they increased their firing rate, suggesting that they were carrying knowledge, right, of the error, right? So we wanted to see, so this is a very kind of heavy-handed, perturbation, right? He's doing this error from the outside. He's sort of driving a microphone, so the bird is hearing this perturbation at a particular time during the song. Maybe we're not surprised that there are kind of dopaminergic responses of dislike, right, when something like that happens to you. Here, what we were trying to do was to see whether dopamine was playing the same role when the bird was, having to evaluate the song itself, right, when it was performing its own natural behavior and converging toward the final song. So what's dopamine doing during juvenile learning? So the way we do this is we look at particular syllables over the course of learning, and then we apply methods to, as we saw in the beginning, the way we parameterize song in our perturbed bird. Very similar idea. We decompose the song, put in a low-dimensional space, and then look at how how different song renditions, how far they are away, right, from the target syllable, which is presumably the adult song over time. And then what is dopamine doing rendition to rendition, and how does that vary with the distance of a given rendition to the song? So here's for one syllable over the course of some days. These are examples where the song is closer to the target. These are examples where it's further away. And what you're seeing now is the dopamine signal recorded directly in area X. So what is the dopamine that's coming in? So the ones that are closer, we get this increase in dopamine. The ones that are farther away, we get a decrease in dopamine, just as one would like. Okay, I'm going to kind of skim a bit through here. We showed that that this signal really scales with the distance of the renditions from the target, from the adult target song in this beautiful way, right? So it really is a kind of scalar indicator of how well a given rendition has been performed relative to the adult song. What's more, Alison was able to extract something that we like to call kind of the dopamine vector. How is the song moving in song space, and how does that correlate, with where dopamine is kind of pushing it? And so if you look at each of those renditions and look at what the dopamine level was from rendition to rendition, one can extract the vector that describes how these renditions sort of co-vary, right, with the strength of dopamine, and get this kind of dopamine vector in song space. And what we see is that the song is kind of systematically moving along that dopamine vector over time. I'll skip that. So we can also look to see exactly what signal dopamine is carrying. So the original experiment that Vikram did, when he just perturbed that one syllable, that could equally well be thought about as an error signal, right? If you just get an error signal, it's good when it's not there. It's bad when it is there. However, if this really is a reinforcement learning paradigm, then we would like dopamine to look more like a reward prediction error, right? So what's a reward prediction error? So reward prediction error, if you remember, is the reward you get minus your current belief for how much you should get. And you can write that out, right, in terms of the reward that you get over previous trials. So what do we see in the dopamine? What we would expect is that if it carries the reward prediction error, we would expect this kind of difference, right? You have the reward having a positive weight and subsequent rewards having a negative weight, whereas so we should see this sort of dip, right, bimodal nature of the filter of the weights in the dopamine signal, whereas if it was purely a reward, it only just responds to whatever happens on that trial. So we can look at the data and extract these weights, and we get this really nice, I think, confirmation that in fact this really is averaging over the performance of previous trials and comparing it with your performance on the current trial. So very much in line with a reward prediction error. All right, I'll skip that. I've told you this model is inconsistent with the location of the error feedback. So how do we resolve this paradox? So one clue comes from, again, some lovely work in collaboration with David Packel here at UW. So one clue is that area X itself is able to modulate the amount of variability in the anterior forebrain pathway. So while we think that LMAN is the major generator of variability, area X also plays a role. So one way that we can see that is that there is a dramatic behavioral context in which the amount of variability in the bird's song varies. So when it is in the presence of a female, here you're looking at the variability in the fundamental frequency of one particular syllable. That variability is quite narrow, so it kind of sings a very well-controlled song. When it is alone, it sings a much more variable song, right? So this is sort of the singing in the shower. This is the singing on stage. Yes, right. And what David's lab had previously shown is the ability for the bird to modulate really depends on dopamine in area X. So we have a really strong hint that that's relevant. That leads to a change, whatever this is doing here, leads to a change in the output coming through LMAN, right? So LMAN is the only place that this whole network is providing any input into the song pathway. And if you look at what happens during undirected singing while the bird is alone compared to when it's singing, there's really dramatically different firing patterns in those two contexts. And one of the things that really struck us is that in the undirected case, overall it's more variable, but you see that there are these patterns, right? These high firing rate bursts that you just never see when the bird is singing the directed song. So when I say we, Michael Faries, my amazing postdoc, proposed that this might be the key, right? That the way that the error signal is being kind of passed downstream to the learning pathway, to the HVC to RA pathway, is via these bursts that are coming through LMAN. So what he suggested is that these bursts may be the thing that kind of open the window for plasticity in this HVC to RA circuit, right? So that LMAN itself, is acting in a reinforcement learning in this rather indirect way, right? In that it's getting input from the dopaminergic signal system that is changing the nature of its firing to go into this burst mode, which then opens the window for plasticity between HVC and RA, which I think is really a really lovely idea. It's a really early variant of what's become, quite a common thing to think about in computational neuroscience, the idea of kind of a 3-factor learning rule. But here, this third factor is coming via this rather indirect change in the firing patterns of this extranucleus. All right, I think I'm done, even though I have a lot of things I'd still love to talk to you about. Right, so let me just skip through this. Just wanted to shout out again, this is with Alison's thesis work. She demonstrated, along with Agata Budzilo, this was really beautiful work done in ARIAX, where the two of them, where Agata in her recordings discovered that there seems to be a cell in ARIAX that no one had ever known was there, that was firing periodically, that seemed to be driving many of the outputs of ARIAX, and that had this really weird firing pattern where it got inputs that had both a positive and a negative input super close together. When you apply dopamine to that circuit, they decouple, so you only get the positive input separated, or maybe it was only the negative input separated from the positive input. Alison had, I think, the brilliant idea that this could really change the nature of the outputs. Don't have time to go through this. I'd love to refer you to the paper, but she showed that this could generate a microcircuit motif controlled by dopamine, whereby, because essentially of the phase response curve of this microcircuit, in the presence of dopamine, this would lead to all of the downstream neurons firing aperiodically and asynchronously, whereas if you switch it with dopamine to having this motif where you only get an input, a positive input, instead, all of the downstream neurons fire periodically and synchronously. So she sort of identified a really cool cellular mechanism for dopamine to be able to switch the output of this network from aperiodic and asynchronous to periodic and synchronous, thus driving the downstream network to be more or less variable. Right. All right, so that was super fast. Yeah. We do have data now from LMAN, so we're working on trying to analyze what's going on there. I just want to, we're really inspired by Eric's work in thinking how that network may be a kind of variability generator itself, in that it's a balanced excitatory and inhibitory network that's naturally chaotic. And so we're trying at the moment to analyze the data that we're getting from that network to see if we can understand the structure of variability that's coming out of LMAN directly and how it's correlated with song. All right, so what I've shown you is that this system has provided for us opportunities to think about learning at multiple timescales, right? The developmental learning of song itself, but this also long or sort of unsupervised learning of the sequence, how it recovers from perturbations. Some of these aspects we think might be unsupervised, other aspects are clearly supervised. So we've touched on, I'd say, many different aspects of how individual cellular properties and network architectures can lead to very distinct roles for all of the nuclei in this birdsong circuit, and how they may be dialing up or down different learning mechanisms that are happening in kind of all of the regions of this network. So lots of work still to do, right? We need these models of these multiple areas, incorporating information about their cell types, their unique learning rules, and their specific circuit dynamics in order to understand how the variability in the circuits generated and modulated, and how that's used ultimately to drive the motor learning at the output. And I'll leave it there. Sorry, I went a bit fast through some of those things, and just to thank again the birdsong group.
Amazing. Let's take a couple of questions. I'm going to warm up, actually, with this one. So on your last slide, you highlighted the sort of specificity and on your first slide, and heterogeneity and different learning rules. On your first slide, you highlighted the sort of compartmentalization and the distinct roles. We were with BOM.DOT in a committee meeting this morning when we were learning about models of language production and video production used at AI2, for example, in which it seems like everything is totally homogeneous. There's an end-to-end training with gradient descent based on reinforcement learning of something or other. So how much do you think this hydrogen atom is going to play out as we think about more complex behaviors and really illustrate a completely different organization that's going on in biology versus that which seems to be taking over the world in AI right now?
Yeah, no, that's a great question. The advantage to a hydrogen atom is that it's totally solvable. The disadvantage is it's the hydrogen atom, right? It's that it's a unique system in which we have this exquisite reproducibility and ability to measure everything. So my answer to that really is that we need to explore slightly more complicated systems. So the next step for us, the ideal next step, would be to look at birds that sing more complex songs, right? So that idea that your song is scaffolded by a unique sequence can't really be true if you're, say, a canary, where you sing the same syllable over and over, and then you sing a different one, and then you can go back to the first one and sing it as many times as you want. So there must be quite a different architecture, even in other songbirds, that would be a very natural next step, right? What scaffolds that learning? How does that work? Can we see any of these rules at operation there? The other place we're looking is in mammalian basal ganglia, right? So we have a collaboration with Garrett Stuber here at UW, who is beautifully, imaging dopamine in the striatum, the whole field of dopamine, signals that are coming into the striatum, while an animal is doing a fairly simple learning task compared to this kind of motor output, but one that one can imagine adding more and more complexity to. And there we would like to understand if the kinds of results we're seeing from our dopamine signals look anything like what's actually happening in striatum, or if you have sort of compartmentalization. The way people typically think about dopamine playing a role in mammalian learning is that there are, this seems so unlikely actually, but that there are kind of pools of neurons that are responsible for going or not going on a particular movement. And that sort of says you've got this very complex code for every kind of movement that you want to do that's sort of, mediated by some small subset of neurons that are now modulated in their learning by dopamine, which seems very hard to believe, honestly. But so what we're going to be able to do is in his learning, in the learning paradigm we have with him, we're going to be able to both see the firing of the striatal neurons and the dopamine signals, which is really going to give us much more insight into how all of that population are behaving over the course of a learning paradigm. It won't be a sophisticated, complex motor learning, but something a bit more kind of low dimensional, but still, I think that's where we're going with that. I don't, obviously, we're not going to get to the complexity of human language learning from this starting point. I think that's kind of a bridge too far, right? But I do think we can get to the biological mechanisms of dopamine-driven learning in striatum.
Incredible. Yeah, thank you. Alex?
Yeah.
So I'm really fascinated by this idea of the variability within the birdsong, or of the birdsong within like a day.
Right.
For instance, right? Is there? It seems like a question that goes back to like, why do they even have a bird song?
Why do they?
Like, why do they, you're spending a lot of biological energy on.
Right.
Like, is there a reason why we would expect, them to hone a bird song?
Yeah. So they use it to attract their mates, and you might ask, even in the difference between directed and undirected song, the song that they sing when the mate is there compared to when they sing that alone, do the females even care about that? And they do. If you play them the undirected song compared to the directed song, they preferentially will pay attention to the directed song. So they care about the males singing that song in this very precise, correct way. So that maybe is the big picture of why.
Do they have the same song?
No. So every bird makes a slightly different song. So, the one thing that we were asked in this paper is, what could we compare the song with, say, the tutor song? So the father will teach the baby his song, but every bird will deviate it slightly. They make a little bit of a change. So every bird has a unique song, even though they all clearly fall within the envelope of, you can tell, if any of you are birders, right? You know what any kind of, a zebrafinch song is, what a canary song is, right? They have a very well-defined kind of grammar and style, but every bird has tiny variations in the song that are unique to that individual. I think that you asked me another question as well.
The question of variability of
Yeah, so why do they vary? The key thing, right, that we really believe is happening is that you need to actively maintain your song, right? You learn it once. If you don't practice and if you don't change it up a bit, it gets worse over time. So if there's any damage to your system and you haven't done that, continuous practice, then you decay, right? So if you deafen the bird and they don't get any feedback, this intrinsic learning mechanism that's always online, that's always maintaining the quality of the song, keeps going. And so what happens is that the song actually gets worse and worse over time. So that is a demonstration that they are actively using the auditory feedback to kind of act against, right, the intrinsic variability to hold the song where it's supposed to be. And so we really think that the variability is a key aspect of keeping the song, well performing throughout its lifetime, even as the physical plant, as the bird is aging, is changing, it's still able to produce kind of exactly the same song over the course of years.
So, Alex, let's take one last question from Ria, and then take the rest of the questions offline.
Yeah, I would really love to know more about how birds are sort of getting this feedback that certain syllables or certain notes are wrong. Are they comparing with the birds around them, or is it
Right.
more inherent?
It's such an interesting question, and one that no one actually knows, right? So what our work demonstrated is the bird is genuinely using an internal template, right, to make an evaluation of how it's doing from song to song. No one knows where that template is stored, right? So one imagines that there's some memory area and that you have some internal model of what your song is and you compare what you do against that. It's a total mystery where that is and how that is implemented. But I think the work really suggests strongly that that's true. And what is a bit unique about this work, there's one other paper in the mammalian system where they also look at dopamine and how it varies during natural behavior. Almost all experiments, apart from this and one other, are done where the error signal is something that the experimenter, that the animal is making some action and there's some error because of something external in the environment. What's really beautiful about this is that it's the internal critic, right? It's that voice in your head that tells you when you're doing well or doing badly. And so much of our own learning it happens that way, right? You're not getting feedback from the environment typically about how well you're doing or how badly you're doing. You have to make that judgment yourself, and most of the basic learning we do really is internal. And I think this is just a really lovely example of seeing that the classic ideas about what dopamine is doing and carrying this reward prediction error really kind of spill over, right, to this internal case. And the only reason we can show it so cleanly is that we have this extremely stereotyped behavior.
Well, you still deserve some external feedback, so why not?
Thank you.